Hi, everyone, and welcome to Creative Biolabs live webinar series. To start, I would like to briefly introduce Creative Biolabs to all our attendees today. Creative Biolabs is committed to supporting life science research clients worldwide through our extensive global network. We are pioneers in providing a comprehensive range of advanced technologies and products in the neuroscience research field. Our offerings include high-quality antibodies, proteins, cell lines, cell culture tools, and modulators. Additionally, our customized services address every facet of neuroscience research to meet your specific needs. With our integrated global resources, we aim to offer you a cutting-edge platform that can significantly accelerate your neuroscience research. Now, today's webinar's topic is the spectrum of stem cell-based neuronal models and their fit for purpose. New technologies in stem cell biology and bioengineering have largely contributed to the rapid development of more complex and sophisticated models of the human neural tissue complex. Stem cells possess a wide range of utilization, from engineering specific human neuronal cell types to the generation of mini brain-like organoids and bioprinting of 3D neuronal networks. Now, these advances in technology enable new and exciting opportunities in the modeling and study of the human nervous system, such as, for example, building of screening platforms for candidate therapies to treat neurological diseases. Now, in this session, we are honored to have Dr. Mirella Dottori to walk us through the spectrum of stem cell-based neuronal models and their fit for purpose. So let me introduce our speaker for today, which is Professor Mirella Dottori. She is a professor within the School of Medical of Indigenous and Health Sciences at the University of Wollongong in Australia. Her research combines technologies in human pluripotent stem cell biology, neuroscience, and bioengineering to develop advanced cellular models of the human nervous system. Her studies have contributed to progress in human neurodevelopment and made an impact in regenerative medicine for neurological diseases, particularly in regards to Friedreich's ataxia. Now, before Dr. Dottori begins her presentation, I want to quickly remind our audience that we will have a Q&A session at the end of the presentation. And if you have any questions during the presentation, please type your questions in the Q&A panel, and then Dr. Dottori will get to them after her talk is finished. So now let's welcome Dr. Mirella Dottori. And Dr. Dottori, thank you so much for presenting your important findings to us today and for making the time to be with us so early in the morning there. And you may begin when you are ready.
Thank you, Sarah. I will just share my screen firstly.
Sure.
Share, and I will put presentation mode. So that's all good?
It's great.
Great.
Thank you. Go ahead.
Okay, thank you. So firstly, thank you to Creative Biolabs for this opportunity to give this webinar. It is an honor and a great pleasure to have this opportunity. But before I start, one thing I'd like to do is firstly acknowledge the traditional custodians of this beautiful land where I'm giving this presentation from. This land is called the Darwell country. I'd like to pay my respects to the elders past and present, and also extend this respect to whichever country and land you're hearing this webinar from. So also just a little bit about Wollongong, which is the city that we're giving this presentation, and from the University of Wollongong. So Wollongong's located about 53 miles south of Sydney. As you can see, it's along the coast and beautiful coastline. Wollongong it comes from the word Wolyanga, from the Aboriginal word, which means the 5 islands. So we've got 4 islands on the coast and the main island. It's also called Ground Near Water or Sound of the Sea. And our university is just, you can't see it on this picture, but it's about an hour's work from the coast. So I encourage you to, if you ever visit Australia, to come by and say hello. So firstly, the theme about this webinar, so the spectrum of neural stem cell models and their fit for purpose. So we think about what's really driving the development of the model, you can really think about it in 3 components. Obviously, there's the why, what, and the how. The why is very obvious in many ways. We think we need the model for understanding neural tissue, disease modeling, drug screening platforms, so forth. However, one thing I'd like you to appreciate from this webinar and beyond is really a lot of our efforts in model is about the development of the model and the technology, developing the tools and the platform in order to then have the model in place to then apply it. And often we're tinkering around in this technology development space that a new model comes up that we can then identify a new fit for purpose. So whilst it's top-bottom, often it's driven also by bottom-up, and hopefully you can sort of appreciate different examples that I'll highlight throughout this webinar in this. So firstly, why develop in vitro models of the nervous system? Well, in my opinion, it is very, very simple because neurons look beautiful. If you look down the microscope and see these neurons, they're just absolutely gorgeous to look at, especially when I was a student or back in the day in the lab when you had a bad day of experiments not working and you look down the microscope, you see these gorgeous neurons, you think, oh, this is why I'm doing this. And I think that's what's also driving my students through the challenging times, and especially also when they're stained, they come up even more beautiful. But on another note, really, when we think about modeling, one of the key components, obviously, is what cell types. So many of us now work in the space of having models that involve human pluripotent stem cells. And again, it's quite obvious why we need those to model human tissue. However, the human pluripotent stem cells have really only been around in the last, say, 25 years. And it's definitely within my lifetime. It may be within some of yours or maybe not. But really, a lot of our models are built from decades of research, which are derived from primary neuronal cell cultures isolated from animal tissues, and obviously also tumor cell lines, which have been differentiated to neurons. And a lot of the technologies and tools we work still to this day are based on all those technologies that were developed with these other model systems. And it's also interesting to think about how and why, what was driving these model developments, particularly when you had the animal there that was accessible. And I'd like to bring you back to one of the earliest studies when they started culturing neurons, and this was by an embryologist, Ross Harrison, and he started the very first sort of efforts in isolating neural tissue, and this was isolated from frog embryos. And it's really interesting to read this from his paper, which said the immediate object of following experiments was to obtain a method by which the end of a growing nerve could be brought under direct observation while alive, in order that a correct conception might be had regarding what takes place as the fiber extends through embryonic development from the nerve center out to the periphery. So it really highlights that a lot of our work is driven by observation and curiosity. And then it's from those observations, curiosity, that the application then comes out. So again, it's technology driven. Firstly, we say, can we do this? And if we can, yes, then of course, there's so many things that we can apply for it. Now, almost a century later from this, we're essentially doing the same thing, but now using human embryonic stem cells. So the first human embryonic stem cell lines were derived in around 1998 from Jamie Thompson's lab. And again, it was very soon after that we were able to start to differentiate to the neurons. However, the first efforts of this was just letting them to spontaneously differentiate, where they can actually observe these neurite processes grow out in the dish. And this was a Nature Biotechnology paper published in 2001. This really excited the field, even though obviously it was tied in with some ethical concerns from this. However, we thought, oh, now here is an opportunity of culturing human cells from a cell line, which is an ongoing resource. And within less than a decade, one thing that I'd like to highlight is that from these studies, obviously all the years of work from understanding neurodevelopment from other animal systems, we could immediately apply this to our human model systems. So one of the earliest work where we could immediately start to refine the process, we found that by inhibiting BMP signaling, by adding noggin, we could more directly drive these pluripotent stem cells down the neuronal lineage and prevent them from going down the other germ layers of endoderm or ectoderm, or mesoderm. So it was really, like I said, less than a decade later that the technology of iPS cells became developed in 2006 from Yamanaka. And this, again, I think all of us can appreciate, really, again, revolutionized our whole model system, because now we had a method of where we can generate iPS cell lines from any individual and start to develop various models of different disease states and try to study that. However, back in the day, and still to this day, we still have a lot of challenges in this space. And one of the greatest challenges is to drive the appropriate neuronal lineage. And I think that's what drive me, the research in this field. Coming from a background in developmental neurobiology, we're studying this in the embryo. We thought, okay, how do we now apply this in the culture dish to derive the specific lineage of interest? And one of the lineages that was of the main interest and still at the forefront of this research is deriving midbrain dopaminergic neurons for Parkinson's disease because of the various different neurological disorders that potentially could be treated from stem cells. In particular, thinking of cell replacement therapies is Parkinson's. So there's been many efforts to derive this cell type, but it's still something that I'll touch on later. However, from the iPS cell technology, again, like I said, this has been really exponential, the growth of the model space in the last, say, 20, 25 years. So we're starting from very simple monolayer 2D cultures of mixed populations. And even in this space, we've become much more sophisticated with CRISPR-Cas9 genetic engineering and other tools in place to now try and derive distinct populations of neurons and even supporting cells, glia, oligodendrocytes. And from there, we can start to develop more sophisticated engineered models, both in 2D and 3D systems, because we can better build the individual units to assemble them together. Also, on the flip side, in the 3D system, we started with very simple 3D cultures of neurospheres, which were mimicking early neural epithelial cells. And from there, in 2013, we had the Lancaster's major paper in the derivation of cortical organoids, which again, many are now applying into their own research space. And this has come a long, long way in its reproducibility and what we know about organoid single cell space. And now, even again, in more recent years, we're talking about assembloids, where we can take different organoids and assemble them together to look at different connectivity between these organoids. So whilst we're becoming better and better, making more complex models, something I'd really like to highlight in this webinar is that we still need to go back to basics. That's really, really key to our model development, and get this bottom part right, trying to understand what are the cell types we're working with, what are the progenitors, to better derive the end cell product. And also to appreciate that empirical basic model systems still have really high value in just to understand establishment of basic networks, to look at different sort of systems, looking at neuron-neuron connectivity and so forth, that they're now applying, say, to learning and behavior. So some of the key challenges when we think about in vitro modeling using pluripotent stem cells, and I think that are still have been current challenge, have been for the past and still to this day will continue to be so, is the method of neuronal differentiation. There's so many different methods out there, the different tweaks, and all of these different tweaks and changes definitely makes an impact, makes a shift into the trajectory of the different cell types that give rise in the dish. So that needs to be considered. The stage of neuronal maturation. So all of us try to gear for the most mature neuron in the dish, but again, maybe it's also the progenitor stage, which is key in many disease model systems, and also for developing specific models, and how you get to the most mature neuron. And then, of course, the need for co-culture systems, so building up that complexity. And this is even talking about how we start to assemble neurons with glia, neurons with oligodendrocytes, neurons and all the mix with microglia, let alone the other tissue cell types that they start to connect to, and the need for that. And I think, again, there's always this, do we look at the single cell versus the mixed cell population of all these different cell types? And there's pluses and minuses of both of those, because again, the value of single cell analysis is to really study the intrinsic properties of that cell in isolation. But obviously we can't all work, a cell doesn't function in isolation, just like us humans. You'll need to see the influence of its environment and the other cell types. So how we start to build that into it. So one system that I'd like to give examples from is the work that most of my research is based on the last, say, 10 years or so, is on the sensory nervous system, particularly of the dorsal ganglia. So many people, when they think of neuronal modeling, we quickly go to the brain, the CNS, obviously, because it's sort of where the complexity is. but really there's a lot of need and in-depth understanding coming from the peripheral. And really the role of the peripheral nervous system to me is fundamental to, obviously, to the brain, because this is what interacts with the external world. It basically picks up all the sensations from your environment, then communicates that information to the brain that then feeds back out to the environment. So the neurons that I'm focused on are these dorsal root ganglion neurons. So they're a ganglia of neurons that reside on either side of the spinal cord, and they are a mixed population. And a lot of our understanding of the DRG neurons basically, obviously, has been come to date mainly from animal model systems across different species. And whilst it is highly conserved, there is also a lot of fundamental differences, even between human and rodent. And one thing I like to highlight is there's been a lot of efforts over the decades in how do we define these subtypes. And they've really been defined by different properties. So they're defined by their size. You can have either small neurons or larger neurons, whether they're myelinated, not myelinated, their conduction velocities, excitability profiles, and also the target tissue in which they're innervating, as well as their molecular profile. So based on these principles alone, you can define them in three very major populations, but obviously it's much more sophisticated than that. There's many subpopulations within each major population. So you've got the nociceptor neurons that innervate all your organs, skin, muscle, so forth, that are involved in sensing pain. They're usually small diameter neurons, usually not myelinated, and have unique ion channel properties and conduction velocities. Then you have the intermediate to large sensory neurons, which innervate predominantly the skin and other epithelial cell types. They're mechanosensory, and they're involved in feeling different sensations. And there's different subtypes that differentiate between light touch and vibration and more deeper, mechanically induced sensations. And then you've got the large dorsal root ganglia sensory neurons, the proprioceptor neurons that innervate your muscle and tendons that respond to different changes in touch and force. Now, there's a real need to understand these cells because they're associated with many different diseases and conditions, more than you can appreciate. One of the main ones, obviously, is chronic pain. There's a huge need to develop new drugs that can treat chronic pain, because the ones that are available out there basically work on the CNS that affect the dopaminergic system and can be addictive. So the working on the peripheral DRG neurons is really a key target. To look for novel compounds that target the nociceptors. Then you've got peripheral neuropathies, and this is a massive umbrella terms, and the causes can be either primary or secondary. Some of the secondary can be associated with diabetes, alcoholism, for example, and many genetically inherited neuropathies. Then we've got ataxia, which I will touch on later on. So ataxia means loss of balance, and whilst ataxia's one of the main drivers is degeneration in the cerebellar neurons, but the proprioceptor neurons and also the mechanoreceptor neurons play a very large role in this. And then, of course, you've got aging. So again, when people think of aging, and affects everyone, all of us eventually, where you have increased frequency of falls, people think of muscle wastage, but actually it's the degeneration that starts off in this process that innervate the muscles, and therefore you're not getting that, and also the mechanoreceptor neurons, and therefore you're not getting that appropriate balance and feeling sensations. So obviously the DRGs, there's a need for this, and the human pluripotent stem cells is a great platform to start to firstly study these different properties, to look at differences between human and other species, and also to develop therapies to treat all these various conditions. However, like all of us, when we come to stem cells in the dish, we come with the huge challenge of how we define them. And we really can only rely on two main parts, which is the molecular profile, the transcriptome and the proteomics, and the functional properties. And even the functional properties, we're limited because it's really looking at the intrinsic functional properties that may be uniquely driven or specified in that cell, because it's not potentially connected to its appropriate target tissue or myelinated. So again, that's one of the major challenges. So many times I think of us like engineers. We're trying to recreate that system to model that. So to derive these cell types, we have to go back to our understanding in development and where these cells come from. So the DRG neurons come from an early population called progenitor cell called neural crest cells. So in development, you've got the developing embryonic neural tube that obviously gives rise to the spinal cord and brain. And in the dorsal region, so that this neural tube gives rise to the neural crest progenitor. So they undergo what we call an epithelial to mesenchymal transition. They migrate throughout the embryo and they give rise to all of the peripheral nervous system, as well as other non-neuronal lineages such as smooth muscle, in some vasculature, cartilage, and also melanocytes. So in one way, we're fortunate with the DRG system because one of the first lineages neural crests give rise to very early in development is these DRG neurons. So in some ways, if you're developing the protocol for this, if you can get to crest and then further differentiate into neurons, by default, you would almost get DRG neurons. So that's helped a lot. The development has helped a lot in this space in developing different protocols. However, it's not as easy as that because even in development, there's still a lot that we don't know about neurocrest, particularly in the human. And we know from other species, and in human it would happen as well, that they come out in different waves. So the first wave that comes out tends to be biased towards giving rise to the larger mechanosensory neurons, the proprioceptor mechanoreceptors. And then later stages of development, you get the second wave, which tends to give rise to the nociceptors. So we know there's subtypes of neural crest, but there's still a lot of work to understand what those subtypes are that give rise to these different lineages. So when we developed protocols to generate neural crest and these DRG neurons, and there was other definitely excellent work being done from different people like Lorenzo Studos from Chambers paper and other papers as well that developed protocols for neural crest. Basically, the systems that we use that we still apply to today is the small molecule and growth factor approach. So we know, and again, I'm not going to go into too much details, but we know from pluripotent stem cells, if you inhibit the BMP signaling pathway and activate Wnt pathway, you get them to a very early progenitor state, which we coined as caudal neural progenitors. So they're not going to become CNS because they don't express PAX6, but instead they go peripheral. And it's these caudal neural progenitors that, if exposed to BMPs, it gives rise to these neural crests, which you can then further differentiate into sensory neurons. Now, this protocol, again, similar to Chambers' protocol, served us quite well. We knew that we could give rise to the various different subtypes of sensory neurons in the dish, heterogeneous subtypes. And we know back in the day we're using MEAs, that they were functional. At the time we were using multi-electrode arrays to measure this. It still took quite a while to mature them. We would start to see functionality by MEAs around after a month, but it was still relatively mature compared to what we're doing these days, but it still worked. However, whilst we could move forward with some of our modeling using these type of cells, we're still very frustrated, I use the word frustrated, and restricted by different challenges that we and other groups around the world think we're having at the same time. And one of them was the variability in our protocols because it was fully reliant on small molecules and growth factors. And there was a really nice paper published in 2018 where they worked on used at least 100 different IPS lines and applied for most of those lines, applied the same sensory diff protocol. And basically, when they did profiling of these cells, they found that there was high variability across the different lines and also within the same cell line across different differentiations. The take-home message was that you need a lot of sample numbers to start to get a signature and something consistent out of that. However, like I said, it was what we had at the time and still quite valuable. The other part that was quite difficult was the functionality, how it would take so long to get these neurons to a mature state to show beautiful action firing potential by patch clamping. So when we started to change our approach in some ways in the lab, where we started to think, how can we start to genetically manipulate these cells to fast track that process? And again, the we is the greater we, the scientific community, where many were starting to use neurogenin-2. So neurogenin-2 is known to be a potent neurogenic factor in development, involved in the differentiation of many lineages. And there are already papers coming out where they were using ectopic expression, induced expression of neurogenin-2 to drive lineages of lower motor neurons, dopaminergic neurons, and also the excitatory cortical neurons. We know neurogenin-2 is also involved in differentiation towards a sensory nervous system, in particular towards this first wave of the mechanosensory subtypes. So we applied this to our protocols. Now, we also did experiments in the lab, and this has also been published by other groups that found that if you just take ES cells and overexpress neurogenin 2, you're going to definitely get a mixed population. You're going to get some that are peripheral neurons, some that are cortical neurons, different types of peripheral neurons. And this has been really well studied at the single cell level to really start to define these different lineages. But it still is very much a mixed population, so it doesn't serve the certain purposes that we're interested in. So what we know is that what is key when you start to use this transgenic approach is the when and how much, basically. So we know that we need to overexpress it at the neural crest stage. And so the protocol, again, that we had in the lab to derive these neural crest cells, basically, we would use the small molecule approach to get them into a 3D neurosphere stage. This neurosphere had a mixed population of crest and CNS cell types. But when we plated these spheres down in the plate, the crest cells would migrate out, and then we would remove the sphere in the middle, and we're left with the migrating crest cells. And still to this day, as cumbersome as this protocol is, we still feel like it's our most favorite protocol. However, we're very much limited to the cell numbers that we can derive from this. So we have to, again, decide what type of applications we can apply to this system. But using this, it was at this crest stage that we would induce expression of neurogenin 2. At the time, we're using lenti. Now we've developed transgenic lines, inducible transgenic lines that we can add doxycycline. And again, we still get the mixed population, different cell types. Transcriptionally, at the mRNA level, we get, fairly equal expression of the different major subtypes. But when we look at the protein level, we also see tends to be biased towards more the mechanosensory, the propio and the mechano, but we still get some nociceptor subtypes in there. However, we get a little bit more consistency compared to the full-on small molecule approach. But the other value of this protocol is that it accelerates the maturation process. So already by 4 weeks, we can see beautiful firing patterns in these neurons. And we've profiled the different ion channels that they express, and they express the main ion channels that you would expect in the embryonic DRGs, the sodium and the potassium, and others as well. So we are not the only ones that have done this approach. Of course, there's other labs that have also shown this. And it's really interesting to compare the protocols. So this lovely paper published in Nature Neuroscience and around 2015, they generated neural crests somewhat similar to us, but didn't use where they generated a 3D sphere and isolated the migrating cells and found that they were biased towards the mechanosensory types, but they found that if they overexpress neurogenin 2 at the crest stage, it would greatly enhance this differentiation process. What's interesting is that, again, a few years later, there was a somewhat similar protocol used to, again, derive to neural crest, but also incorporating small molecules. But here they overexpress 2 factors, neurogenin 2, together with brain 3A, which is also expressed in all the various DRG subtypes. But what they found, it was the length of the expression, the timing, of the expression of the transgene, which would influence the end cell type that they would get. So they would get the mechanosensory neurons. However, if they did a longer expression of neurogenin 2 with brain 3A, they would also get a unique other subtype, which was also sensitive to thermosensation, which is very interesting. And again, comparing another paper, again, very similar method in terms of using only neurogenin 2, overexpressing that at the crest stage. However, in this report, there was actually enriching for nociceptor neurons, which is, very interesting, which they did lovely profiling at the single cell level and also functionally. But when you look at the CREST protocol, it's tweaked a little bit different to ours and the other reports as well. And so again, I feel as if when we compare all these different papers, whilst we're all using neurogenin 2 or different components and different timing, I think part of the variation in our protocols is the tweaking of getting these neural crest cells. And I come back to the very beginning that I highlighted in this talk, we're working on this progenitor type and also subtypes of these progenitors, I think is greatly influencing the end cell type that we're getting in our modeling system. And it's something that I think the field needs to work on more. So nevertheless, we can still definitely apply our models to ask different questions and gain new knowledge. One of the main things is comparison of human and mouse knowledge. So paper published in Science earlier this year by collaborator Gary Lewin, does beautiful work in mechanosensation. He identified the novel function of one of the mechanically gated ion channels, ALCON1, involved in basically mechanosensation in touch and feeling. And we were able to validate the extensive studies that they identified in the mouse. We applied a knockdown of ALCON1 in our human sensory neurons and found that there was a reduction in mechanically activated currents as well, therefore further validating the findings in the mouse. Also, there's obviously the application in disease modeling, and one that I'd like to focus on that we work on is Friedreich's ataxia. So very briefly, Friedreich's ataxia is a genetically inherited disease. Ataxia means loss of balance. Usually, not always, but usually you start seeing symptoms in early childhood where children start to easily fall, become clumsy, and over time become in a wheelchair, and the degeneration is ongoing. And it's mainly in particular parts of the cerebellum and also in the peripheral nervous system. Some of these children also have cardiomyopathy and other sort of complications. The occurrence is anywhere between 1 in 30,000, other reports say 1 in 50,000. But one of the main types that do generate in the peripheral nervous system are the proprioceptor neurons. So it's one of the primary neurons that degenerate and is obviously contributing to the ataxia. Now, what's very interesting about this disease is, whilst it's rare, I see it as low-hanging fruit in terms of a therapy, because we know the gene that's involved. So the gene is frataxin. Frataxin is a highly conserved gene. It's also expressed in yeast. It's ubiquitous. I wouldn't say ubiquitous. It's expressed in all cell types of the body, but at different levels. And basically, frataxin plays a role. It's encoded in the DNA, but it functions predominantly as a mitochondrial, in the mitochondria, as a mitochondrial protein, and it is involved in forming iron sulfur clusters, which is needed for complex 1 and ATP synthesis. So what's interesting about this disease is that the mutations of frataxin occurs in the intronic region. It's a GAA repeat expansion. So the bottom line is that these repeat expansions cause epigenetic changes, which affects the transcription of frataxin. So in these individuals that have homozygote repeat expansions on both alleles, they have very low levels of frataxin protein, and that causes dysfunction of the mitochondria. So in terms of therapy, one of the two main ways of looking at therapies is obviously looking at antioxidants to help improve mitochondrial health and homeostasis. And the other is gene delivery systems to deliver frataxin. But it's not as easy said than done, because obviously we need to target the appropriate cell types, because they've shown that too high levels of frataxin can be toxic and can be somewhat worse. So you've got to get that Goldilocks level to get it right. So we and others, since actually soon after the IPS lines were first generated, myself and colleague generated some IPS lines from this disease, and same with other groups around the world. Because of this, we thought, okay, this is a disease that potentially we could, develop therapies for and make a great impact. But at the time, we didn't have the protocols developed for sensory neuron diff. We were really relying on our protocols to generate neural progenitors, which would give rise to more cortical progenitors. We did extensive molecular profiling of the mitochondrial function and other studies. And the bottom line was that we didn't see any differences between the IPS and the controls that we had at the time, which wasn't isogenic control, but other IPS lines. But now, obviously, the field has advanced greatly, and we've gotten better on all fronts. We've now got isogenic control lines. We've got better differentiation systems. And this Neurogenin 2, we found, is a powerful tool to accelerate this differentiation process to apply to different platforms. So we obtained some IPS lines, and they're isogenic controls from the University of Alabama, who was generated by Marek Naparella and Jill Naparella. And we genetically modified them to insert the Neurogenin 2 expression cassette. And we're using this in different different programs, which I can't go into, but watch this space. In terms of showing the data, we're going to just highlight what we're doing. So obviously there's the comparison of the disease neurons with the isogenic healthy control neurons. And at the moment, we're doing extensive transcriptomics, proteomics on these neurons, looking at mitochondrial function in these cells through high throughput imaging analysis as well, and extensive functional profiling, looking at excitability profiles and mechanosensations, their response to mechanosensations. So watch this space in the field. We also have funding from the philanthropic organization to start applying drug screening to look at to different compounds that may improve mitochondrial function. So at the moment, there's only one drug that's approved, FDA approved for treating FA. That was only approved, I think it was last year. It's of omaveloxolone. I'm sorry, I mispronounced it. Omaveloxolone, or also known as Clarence. This is yet to be approved in Australia by the TGA. So now this has opened the door to looking at other drugs that can work in combination with OMAV or in isolation, but working on similar systems. OMAV basically works on the NRF2 pathway to improve mitochondrial health. And then the third arm, which I'll touch on, is basically using these stem cells as a platform to look at gene delivery systems to deliver frataxin. So in this space, we've worked on different systems. So I've collaborated with excellent bioengineers from University of Melbourne. So that's Frank Caruso and Christina Cortez Hugo, where we, in this work here, we actually use cultured our neurospheres in 3D, as small sort of like sensory spheres. And as those who work in nanoparticles would know, you've got so many different varieties and sizes and the way they're assembled and how they're assembled. So we started looking at the layer by layer approach of nanoparticles, and we compared different sizes of nanoparticles, the different charges, the core, whether it was silicon or polystyrene, and we also looked at glycogen. And we looking at GFP, what could more easily penetrate into and be taken up by these sensory neurons. And basically, we showed that it was the smaller nanoparticles that were positively charged and had a core dense silicon base that was the most efficient in basically taken up by the sensory neurons. And we also got very promising results with glycogen. And then we followed this up by, again, modifying these nanoparticles to now deliver frataxin plasmid. We knew that the plasmid we were using wasn't the ideal therapeutically, but it was proof of concept to see, okay, let's now start to incorporate DNA into the nanoparticles to see how it could be taken up. And we got really nice results where we saw increased frataxin expression. So this is now moving forward in this space and looking at different ways you can incorporate, and this is both the biology side as well as the engineering side. So we're looking at different modifications in the nanoparticles, but from biology side, you could think about mRNA as well as drugs as well, and thinking what could potentially cross the blood-brain barrier as well to deliver into the cerebellum. Also in parallel, what we're doing is using our sensory neurons. This is a grant that we got from the Australian Government Medical Research Future Fund, which involves labs across Australia, where we're screening basically different AAV libraries. So one of them is an AAV testing kit, which has got at least 64 different AAVs, which are being used currently in the clinic or being intended to be used. And then we also have 6 different bioengineered AAV libraries, which have been modified that we're screening. And each of these AAVs are barcoded, where we are basically transducing our cells, our sensory neurons derived from our different FA lines, to see which one has the most efficiency in infecting the sensory neurons. Doing this in parallel, testing using the same lines to look at cardiomyocytes derived from these cell lines, and also in collaboration with Samuel Naylor from QIMR here in Australia. He's also grown the cerebellum organoids using these same cell lines. And this work is really coming along really nicely. So again, watch this space. One thing that I also want to touch on is that for the dorsal ganglia system, when we're thinking about the application of sensory neurons, this is not a model as such, but it's still an application, is for transplantation. And we have also done some studies in this space where we've transplanted sensory neural progenitors into the dorsal root ganglia of adult rats. This is technically highly, highly challenging. And I had an excellent PhD student at the time, our postdoc, who's brilliant with mouse surgeries, because the DRGs are tiny and where they're located under the bone, very hard to target. But we were able to show proof of concept that when we transplanted, they did differentiate after 8 weeks into the different sensory lineages of the 3 major types. It is obviously more of a challenge, but it's something that I think in parallel is always important to pursue, because, back many decades ago, no one would have thought reprogramming would have been possible. So I think all avenues always be to pursue, as great the challenge is, that one day we can potentially get there, even though obviously assembling the network is very complex, but you'd be amazed by the capabilities of what the body can do. Well, I am anyway. But one thing that is really key to important is now we're talking differently, because you have to talk about generation of a pure population of progenitors, not the end cell type. And as I mentioned at the beginning, there's been a lot of work done in this space for Parkinson's disease, because for cell replacement therapies, this has been at the front line as proof, because it's shown proof of concept, promising studies where they've used embryonic tissue that could potentially recover some Parkinsonian symptoms, some individuals. However, leaders in the field in this space, including Mile and Palmer and others, did very recent studies showing nicely that even if you're able to get a pure population of homogeneous population of midbrain progenitor precursors in the dish, once they go in vivo, it's a very different environment, and they still have the potential to give rise to other cell types, not just glia, but also potentially other neuronal lineages. So we really need to appreciate that what happens in the dish in vivo, depending on the microenvironment, can still influence the outcomes again, and may not necessarily be a direct translation, and something we always need to be mindful of. Then the final sort of application I want to end on is the development of tissue models. And again, in the sensory nervous system, there's really, I think it's a great cell type great tissue type to work with in terms of neural, because DRG neurons do innervate all tissues, particularly skin, muscle, but also organs such as the nociceptors. And there's some really exciting studies coming out in this space. One that, again, they've published a couple of papers on this, and this one where they showed they made these neurospheres which were differentiated towards the nociceptor sensory neurons. And they're able to encapsulate these in these PDMS gels. And they co-cultured these with, basically they developed skin-like constructs, which they also differentiated to. But the challenge with skin is that you have the different layers. You've got the dermis made up of the fibroblasts, and you've got the epidermis, which is made up of the keratinocytes. And when you're developing in vitro models of skin tissue, the way keratinocytes differentiate is that they need this air-liquid interface. So you literally have to raise the culture to be exposed to this air-liquid interface to develop this keratinocyte layer. So there's that extra challenge. It's not just about putting these cells together. You've got to also manipulate the whole organ structure to accommodate that. And they were very clever in how they did it. They basically raised the skin tissue so it could have this air liquid interface, and showed innovation into the skin layer. Also, this obviously is really accelerating forward in the organoid space, and there have been a couple of papers, obviously not as many as cortical organoids, but definitely still happening for DRG organoids. This is, again, beautiful work done by Barna Broccoli in Italy, where, again, they're working on Friedreich's ataxia, and they made DRG organoids from FA lines and healthy controls and profiled those. And here in this paper, they started to do these co-culture studies where they basically put the organoids together with muscle cell types, which they also differentiated in vitro. And you could see this starting to get these interactions of the sensory neurons. Here's the axon shown in blue with the muscle tissue by EM around it, and start to model differences in the disease neurons versus the control. So in this space, we're going through using a different system where we're looking at bioprinting, because, again, we're toying around with organoids. We're also still interested in these single cell types and trying to assemble them into different stoichiometries and also different layers to neighbor them together. And so one of the values of building transgenic lines such as neurogenin 2 is that you have the capacity to do this in different systems, because obviously neurons are delicate. You can't print neurons, you can't fax sort them, but you can do this with the progenitors, the neural crest. So work that we've recently published was that we were able to use this neurogenin inducible transgenic line to print the neural crest progenitors into a 3D matrix using a JAMA bioink. And once it was in the 3D, we could add doxycycline to induce expression neurogenin 2, which accelerated, again, that maturation process. So we could get the differentiation of the neurons within the matrix. And you could see within this matrix, we've got extensive neurite outgrowth, still retaining expression of the sensory neuron markers. And we saw outgrowth and also glial differentiation, both top to bottom layers. So, and not shown, we also showed that they were functional by calcium imaging. So being Italian, I see this, as like a tiramisu cake where you have a layer by layer, different. That's how I see where we can build different layers in bioprinting of different cell types and to start assemble them together in different functional units and incorporate different, say, microfluidics channels to look at connectivity. So again, this is where I see this work going in this space. So I've been focusing on the model systems, obviously, but it's not one thing that is really important to think about. It's not just the models we have to think about. The models will not go anywhere unless you have the appropriate biological readouts. So as much as we're putting efforts in developing the models, I think it's really critical to develop the tools and the technology in having the appropriate readouts to interpret the models and get the value of it, because the two need to come together for the application of the model to serve its purpose. So, and also on this note, I think what's really important is we're not talking about technologies, not technologies and the tools of the readouts, but also in consistency. So a paper that's very recently come out, just hot off the press, a lovely paper again, is looking at the DRG organoids. And what they did was they did very beautiful single cell and extensive profiling of DRG tissue isolated from human embryos. And they also profiled DRG organoids derived from stem cells. And they were able to compare the different cell types that arise from both and to look at both the consistency and the overlap and also the differences in development. And I think this is really, really key because, again, with the technology of single cell sequencing, and which, like I said, that's one of the main ones that we have to rely on to validate our models, particularly derived from stem cells, because it's out of context of the surrounding tissue. So we need a lot more in that space, as well as functionally. We really need a better, there's papers coming out in developing different pipelines of multi-electrode arrays or even patch clamping signatures, where we can start to develop a signature of these cells, both in vitro to model that to the in vivo. And the other thing, the paper that also recently came out in Nature that I think is really fundamental and speaks to all of us is where they started to collect all the data sets, and this is cortical organoids, and they compared organoids, I think it was from 29 different papers and data sets, and to look at commonalities between all this transcriptome and compared that to what's known from the human brain atlas tissue. So we can start to benchmark how our protocol is compared to everyone else's protocol. There's no right or wrong. I'm not saying this one's better versus the other one, because I think variations in the model is really, really important too, to start to understand what these differences may be. So again, I come back to, whilst the why is critical in driving the development of the model, a lot of it is in the technology development on all aspects, not just the model itself, but also the tools. So the main take-home messages I hope you got from this webinar is the importance of the progenitor. I've been focusing on the neural crest, but I think that goes across the board of all different lineages, that you really need to put a lot of, not you, need to put a lot of effort in understanding our progenitor population and the diversity. And as we get better with lineage tracing studies, with barcoding to say this progenitor is going to become that end cell type, we're going to develop better tools and protocols in place to derive the lineages that we're interested in. benchmarking our stem cell derivatives. And this is not just back to the human tissue, which we're getting better at, but also amongst the different labs and different groups. And the final one is sharing the resources and data, because really, like I said, there's no right or wrong. It's really when we share our tools and our protocols and the data we get out there, even if they're outliers, even if they're different, I think there's huge value in appreciating why did this become this and that didn't, because we need to serve that as our advantage. So I end with the quote that it takes a village to raise a child. I say that it takes a village to develop a model, village being the whole scientific community. And this, again, a lot of this work that I talked about, our own wasn't possible without all the funding from various sources and collaborators. I just put some here and the organizations. And I'd like to thank you for your attention.
Thank you, Dr. Dottori. That was a wonderful presentation. Let me quickly share these slides again. Yes. Yes, so I hope our audience found it as engaging as I did. And so now, without further delay, let's start the Q&A session. Let me quickly see, sorry, just a technical. Yes, okay. Sorry. Yes, so let's start the Q&A session. So we have a couple of questions from the audience already. So first of all, do you think an NGN2-based approach will be appropriate for all cell types? For example, the INDI project at the NIH has protocols for cortical and motor neurons, but as compared to traditional growth factor approaches, NGN2-induced neurons have some differences. So in case you are familiar with this work, do you think these differences could be addressable with modified protocols?
Yes, thank you. Oh, sorry. Yeah, thank you for that question, Charlotte. I agree. I think that there are definitely differences in the protocols, and I haven't studied, those protocols as carefully as I have been in the sensory neurons. But as I flagged earlier on, I think one of the key, whilst a lot of the efforts, even including from our lab, we've been focusing on the transcription factors, the combination of the different transcription factors to drive those lineages, and the timing of how long they need to be exposed. Again, I believe that a lot of it is in the tweaking, and even if the protocol isn't involving a specific progenitor type, such as, like in our case, we need to get to neurocrest and then move on, it really is a spectrum, like a road, because it's adding different concentrations of these small molecules at different timings that is really giving rise to the progenitors there, and it may be a heterogeneous population of progenitors. So again, I think tweaking that part of the protocol is just as critical as the transgenic approach, even if the transgenic approach has been refined really well. Because even for cortical neurons, they need the small molecules at the beginning to then switch on neurogenin to get, the deeper layer cortical neurons, as opposed to just throw neurogenin too. Hope that answered that question sufficiently.
I think it does. So let's move on to the next question. In co-cultures, how important is it that iPSCs have the same origin? For example, are they supposed to come from the same donor? And is it better to have a single donor or to vary the donor to get the best differentiation for each cell type, which may then involve multiple donors?
Yeah, that's an excellent question. Because we think about that with our Friedreich's ataxia lines, obviously. So obviously there is great variation between different IPS lines, even healthy control, because at the end of the day, they are epigenetically modified. It's reprogramming. So you're going to get variations on that level of reprogramming, and which may influence the differentiation process. I'm on the side of reproducibility. So I think it's of value to have different IPS cell lines and to see that variability. And if, despite that variability in the cell line and from different genetic backgrounds, you still get the end result you desire, I think it's actually a strength. Because one thing that I didn't mention in the talk, but I often think about, is why are we trying to develop these models? Really, why are we trying to develop it? And it's not just for us to do that experiment in our lab. For me, the huge value of a model is that if other labs can then copy it to apply it to what they want to do. So I feel like reproducibility is a high priority on the list, and to make it more streamlined and easy for people to use. And that was one of our rationales to generate the Neurogenin 2 FA lines, so that we could share that resource with other people within the community, so that they can more easily generate the sensory neurons or the neurons of interest, because rather than going through a more lengthy process. So while we're doing this, they can move on and do that. So to answer your question, I wouldn't focus too much on, I personally wouldn't focus on trying to get it perfect, having the right line from it, because I think, again, if you can show that no matter what, you still got that end result with the co-culture, a muscle derived from this line with the neurons derived from the other lines, it still worked, I think is actually a strength.
Excellent, thank you. So we have another question from someone else. What are the best ANN, so artificial neural networks, to train these organoid neurons?
Yes. Best, I wish I knew. It's a great question, because it's something I've been having conversations with others about, and something we're very interested in working on in that space. When I say that, okay, that it's a really good question, because they are different. I'm a developmental neurobiologist, so I am biased in the type of neuron that you place down, even though I also, it's not a scientific, sort of coming from a scientific, but I feel the neurons are flexible as well, because in the dish, they will behave differently to in vivo, like I said. So even though it may be a sensory neuron, it could potentially behave more like a cortical neuron given the particular environment. Who knows? Who knows? I'm just putting it out there. Some things are intrinsically driven, some things extrinsic, and some can be modified. So to answer that question, does it matter the cell type? I think to a certain degree, but not, it would be advantageous to work with the neurons that are predisposed to becoming that neuronal cell type for that purpose. All neurons need to learn, all of them, even the most basic neuron. And my preference to the peripheral nervous system for learning is because they are the first neurons one of the first neurons that need to respond. I keep saying this to everybody I speak to. When a baby is born, you hold them, they need to feel the ground underneath them to walk. They need that feedback. They don't know how to work. You don't tell an animal or baby how to walk. You put this leg in front of the other. They're feeling it. It's that feedback from the peripheral nervous system. These neurons are learning how to innervate the muscle, how to respond to that tactile information, and how to respond appropriately. They're not thinking of maths equations or language and so forth. So a lot of our basic intrinsic learning, we're still like thinking of muscle memory. Is it really the muscle that has the memory, or is it the neurons that are innervating the muscle that has the memory? But everyone's focused on the muscle memory. That's why you're doing things automatically, like driving a car without thinking about it. So I think one of the best neurons to show learning and networks would be the peripheral, and also, compared to the cortical, they're bipolar for other reasons. But that's maybe because I work on them as well.
Excellent. Thank you for that elaborate response. Regarding the nanoparticles, how do you envision them to be translated into therapy? Although this was maybe a proof of concept, but how do you envision that they could maybe specifically target the sensory neurons specifically and not result into any off-target effects?
Yes, so that's an excellent question. We, just like for the AAV work, ultimately these need to be tested across different platforms, as well as obviously animal models. But one of the powerful things about nanoparticles, which I didn't mention, is that they can be modified to express cell surface markers to target those cell types as well. And so if we know of unique receptors expressed in these neurons, which we do, they can be modified. That coupled together with the delivery system, knowing doesn't it cross the blood-brain barrier, there's more capacity to tweak I feel, the nanoparticle as opposed to, say AAVs. Saying that, nanoparticles, you also have the challenge of trying to get it into the nucleus if it needs to go into the nucleus. So it's an excellent tool, I think, for cytoplasmic expression. That's why it's gone quite a way with mRNA. But when it needs to get into nucleus, that's where the AAVs obviously may be a better tool. So it depends on the need, the purpose, but definitely still valid because there's a lot of work being done with mRNA delivery, ASO deliveries, as well as drug delivery as well. So like I said, in this space, you've got the biology, we're thinking, what do we need to deliver? Working together with the delivery system. And just like any delivery system, depending on delivery system, you need to modify the biology to be better adapted to that delivery system and vice versa.
Great, excellent, thank you. And then we have one final question. I think, so the question is, what type of cell line can be used for a neurodegenerative disease, Yusuf? So I think this will probably depend on the type of neurodegenerative disease.
Exactly.
And what your goals are, but yeah, feel free to elaborate.
And also any cell line, truly any cell line. If the human embryonic stem cells or healthy control iPS cells are really powerful and in some ways easier because you can genetically modify them to introduce your mutation. You can add different factors in the dish to model what's going on, add cytokines, inflammatory mediators to model the disease pathology. So you don't need to have potentially, be of value to have that disease iPS line, but it's not critical. You can still move on with understanding neurodegeneration with even a tumor cell line. Even, like I said, I was thinking when I did this talk, I'll end with, I grew up without internet. People think, oh my goodness, how could we survive without internet? But it is possible even without the human pluripotent stem cells. There's definitely a lot of capacity using other lines and other tools to make meaningful results.
Excellent, thank you. That's a great answer. And then with that, considering, our time is up. So I would like, again, to remind everyone that Creative Biolabs regularly invites esteemed experts in life science and biotechnology to share their latest findings, such as Dr. Dottori just did in our webinar series. And we encourage everyone to visit our website and subscribe to our social media channels so you won't miss any upcoming events and future webinars. And that's it for today. So thank you everyone for joining us, and especially, again, Dr. Dottori, thank you so much for this very insightful presentation and Q&A session. That was really excellent. We learned a lot from you. And so we will hopefully see you all next time. Thank you.
Thank you. Thank you everyone for your attention. Thank you.
Thank you.
It was a pleasure. Thank you.
Likewise. Thank you all. Bye.